thanks a lot, Jakob, for choosing to take on this request at a very, very last minute. But I think it's a nice forum for people to be able to see your work and for you to share your research. So thanks a lot for coming or for engaging. Apologize if you guys can all hear my son playing a video game in the background. So I'm going to give Jakob a brief introduction. I've known Jakob's work for a while, and I'm sure a lot of you guys have too. He's written some really interesting manuscripts in the last little while that appeared in Neuron, and he's a co-first author on a science paper that I suspect we'll be talking about today as well. But I won't give away, any of your great stuff. I'll let you do that. But I'll give you a little rundown of who he's trained with and where he's trained. So from 2013 to 2015, he worked as a postdoc fellowship, which is this really unique kind of in-between stage at the NIH with Leslie Ungerleider, working with fMRI and structural MRI processing in the context of non-human primate work. And then did another unique kind of training stint at where he did half of his PhD at Cambridge and half of his PhD at the NIH with Ed Bullmore and Armin Razanahan, respectively, looking at population statistics and novel metrics on allometric scaling and total brain size variation, inter-individual variation, and then the creation of morphometric similarity networks, which is kind of one of the things he's definitely best known for. And for the last 2-ish months, he's been a postdoctoral fellow with Aaron Alexander Block and Raquel Gur, so a great time to be starting a postdoc. He's been where I guess we'll be working on integrative approaches for personalized medicine, and kind of taking a real multimodal approach with single-cell RNA sequencing, histology, and looking at connectivity. So thanks a lot for taking the time to do this in such a weird moment. And I am, among many others here, are looking forward to your talk. So go for it.
Yeah, thanks, Molar, for inviting me. It's, I guess, a really nice time to be able to get anyone from around the world to speak. So as Molar mentioned, I just started my postdoc, so most of what I'm talking to you about today is kind of a product of my PhD. And so it centers around imaging-first approaches for integration with mostly the Allen Institute dataset, which I'll get into in a second. But to kind of frame things to start, I wanted to just give an idea of where all of this was motivated. And so Haeckel's kind of, contentious drawings, disputed drawings, still demonstrate a very interesting concept in speciation, which is that at various stages of ontogeny, the human looks very similar, and therefore there's conserved features of development that are reflected through the evolutionary process and then played out during ontogenesis. And to revise that a bit and to move it forward in terms of how constraints on variation kind of manifest in humans, we can see that gastrulation, the transition from single-layered blastocyst to multi-layered gastrula is a conserved process of ontogeny across phylogeny. However, there's clear other areas in human-specific ontogenesis where potential explosions of variation might have occurred relative to other species. And when we're looking at neuroimaging, it's really important to remember that these constraints play out in terms of the search space for the variation that we're looking for. And one specific example is that across primate evolution, cortical folding has become extremely complex, and the cortex has become very gyrified, and certain aspects of cortical architecture are actually quite conserved in terms of magnitude differences. And this is kind of best articulated by some work by Bruno Mota and Susanna Huctilano-Hosel, showing that the complexity of cortical gyrification across both lissencephalic and gyroencephalic species is a product of the total surface area expansion and the square root of the thickness differences. And so what's interesting is B here shows actual magnitude differences in cortical thickness in absolute terms versus absolute total surface area on this plot, and you can see just the magnitude, the order of magnitudes greater in terms of surface area. And when you get to this point of 1.2 millimeters cortical thickness, for some reason, gyrification or gyrencephaly is kind of emergent. So that complexity of this large scale temporal impact on cortical microstructure is somewhat played out in development. And I think that this is also really echoed by some early work by Lyle and colleagues showing surface area versus cortical thickness expansion right after birth. And birth is a really interesting time in ontogenesis because relative to other species, we're very premature. Horses, when they're born, can stand up. Babies can't even hold their heads up. And so this is shown, it's very small, but the percent change in terms of cortical thickness and surface area are just drastically different. And so these concepts of, population variation versus inter-individual variation that I'm going to be showing you in terms of single brain maps are really important to contextualize in terms of the underlying neurobiology. And so luckily, these parallel fields of psychiatric genetics have spearheaded kind of large-scale consortia to kind of examine psychiatric genetics at a massive scale, but also in terms of the categorical versus dimensional distinctions or lack thereof between conditions, which are, a lot of them, emergent in terms of their phenotypes in humans. And so the main data set that I'll be talking about today to bridge across these scales is the Allen Institute data, which is relatively recent in terms of neuroimaging. It only kind of came into kind of the public view in 2012. And since then, some really beautiful work from these consortia showing trajectories of these different features of genetic architecture and cell types specifically, as updated recently in this paper, kind of demonstrate the dynamism that occurs before birth and also after birth, and also not shown here between species. And so the idea to do all of these mappings between the data sets was definitely not mine. And in kind of the biggest kind of paper that spearheaded a lot of this was the Ricciardi and Altman paper in Science, which I'm sure many people are familiar with, and it examined functional connectivity relationships to gene expression architecture. And since then, I just wanted to highlight two resources that I'm not going to be using or did not use in my studies, but are absolutely fascinating resources. Arena's paper in 2019 on how to kind of map the Allen, the considerations and the different steps, I highly recommend reading. I won't go into many details on these pipelines today, but it's something that we're now integrating with, and I highly recommend looking at that, and it's very flexible. The other one that I kind of stumbled upon is these vertex-wise maps of gene expression, which are interpolated from the Allen Institute data. And so this provides kind of an interesting opportunity debated about because of the actual resolution of the data. But this was a cool resource that I just stumbled upon recently, and I'd like to highlight here. And if you read this paper, they have their data available online, or you can email me. So generally, today we're going to be just kind of traversing the biological hierarchy, if you will, not kind of literally, but just based on the relationships that we're measuring through correlation and other methods between systems level architecture at the macro scale and imaging, and potentially genetic architecture. I would argue gene set architecture, so combinations of genes. And so the first study I'm going to talk about is population-based, and it's a single map of cortical scaling of surface area. So a single feature that I was introducing as incredibly important and kind of emergent in terms of variation across evolution and development in humans. And so the second one and the third one will center around individual multimodal based approaches to looking at brain organization. And each of these will represent a different way of integrating with the Allen Human Brain Atlas. So let's start with the scaling. So basically, the motivation for this project was the fact that brain size variation is almost, it can vary almost twofold across human adults. That number is slightly debated, but generally about twofold. And so that's a fascinating construct given the spatial focus, this focus on spatial statistics in neuroimaging, because how does this variation across kind of canalized adults or echoes of development that are more or less finished in terms of time, how does that represented locally? And so this was the methods were motivated by surface area expansion studies in from David Van Essen, and in humans relative to macaques, as seen on the top, with local expansion being preferentially in heteromodal association regions, and similar patterns between babies and adults in terms of surface area scaling, as shown in B. And so to kind of complete the triangle, we wanted to ask, as human brain size variation emerged across different individuals, what was the local effect? And this idea, again, was not new. It's based heavily in allometric scaling and functional asymmetry literature, and the framework for evaluating these scaling concepts is a log-log regression framework, where you look at one piece of the whole relative to the whole, or even one piece versus another piece of an organism. In this case, the kilo length versus the body size in these fiddler crabs. And in humans, you can see from this graph that different organs are not scaled equally as some simple constructs such as body size increases. And so this motivated us to look at brain size in humans as measured by total surface area relative to local surface area. So schematically, what this looks like is a map of isometry versus hypo or hyper allometry. And in this case, these log-log coefficients above 1 would represent hyperallometric scaling, or as a bigger brain, as a brain size increases, the local area disproportionately increases. And hypoallometric scaling, which as brain size increases, the local surface area disproportionately decreases, but it's not actually a decrease. It's still actually an increase, but less than isometry. And so to do this, these ends were big at the time of conception, but I guess not anymore. But there are 3 independent studies processed a bit differently. One's longitudinal in terms of the modeling, that's very important. And we replicated in the Human Connectome project data across different smoothing kernels. And so these were what the maps looked like. And across data sets, there seemed to be a convergence between regions that allometrically scaled relative to total size variation versus hypoallometrically scaled. And you can see that there's obvious differences in the conjunction map between the PNC and the NIH data set, but by and large, there was a significant relationship in these independent studies. And this was also replicated to a slightly lesser extent in the HCP, most likely due to processing differences. And so to annotate this map, we were very interested in using the Allen Institute data, and were inspired by the Ricciardi methods that I introduced before. And so the Allen data is comprised of 6 different donors, 2 of which have full brain coverage, or more or less full brain coverage. The others only the left hemisphere, for methodological purposes as well as financial purposes. And basically, you can see that the sample distributions vary across the different donors. And so to align our data, what we did was we took our imaging data on the vertex, at the vertex level, and we matched the closest sample locations in the Allen Institute space with a 10 millimeter radius in terms of the imaging averages, and we ran a linear mixed effects model to look at the spatial relationship between gene expression and imaging phenotype, scaling in this case, and where we could include donor as a random effect, and therefore do our linear models across the about 2,000 samples. And therefore our N effectively was 2,000 relative to the 10,000 vertices that we sampled in the imaging. And so you can see here that I wasn't going to really focus on the enrichments, but basically the genes that showed most spatial correspondence in terms of their expression profiles relative to these cortical scaling maps. Well, first of all, it was highly replicable, and second of all, these large, these kind of widespread terms in the ontology kind of popped up consistently. They're very much related to metabolism and neuronal structure. And so the second kind of method was kind of orthogonal to what I just presented in terms of population-based statistics. And we called them morphometric similarity networks, effectively just looking at individual brain organization, in this case in a normative population. And since it was a network-based approach, it called for different integration strategies with the Allen Institute data to annotate our findings. So this was motivated by looking at the various cortical morphometrics and morphological estimates that you can derive, the gray matter volume and its constituents, thickness and surface area, as well as local estimates of curvature. And this kind of canonical study, which showed integration of various features, resting state fMRI, functional fMRI, and structural components in terms of parcellating the cortex, was kind of coming out at the time. And we were wondering whether we could generate some related method to look across different cortical features in a network-based approach. And so this was also in part motivated by histological studies showing that cytoarchitectonic similarity was the best predictor, along with distance, in predicting whether there exists a connection physically between two regions. And this was great work done by Alex Goulas in Klaus Hilgetag's group. And recently, there have been really two great studies that have more explicitly examined this question about cytoarchitectonic similarity by looking at intensity profiles by in big brain, the 3D reconstruction, histological reconstruction of a single human brain, as well as in vivo data sets like the Human Connectome project. And what I was kind of thinking of in terms of the morphometric similarity was based on this early quote from Brodmann, which is the idea that it's not just one feature of the cortex that contributes to functional sophistication, as he calls it, or kind of the substrates underlying function are much more complex than just a single feature. And so I would argue that this triangle would kind of more or less comprise the functional sophistication, as Brodmann kind of mentioned it early on, and that what we were trying to kind of fit in terms of methodological considerations was a way to get cortical similarity in terms of the microstructure. And the reason why I'm saying morphogenetic is because we've showed that these MSNs are related to gene expression. And so to do all of this, we used two normative samples, an overlapping study, as I mentioned before, at the NIH, as well as a collaborative study between Cambridge and UCL. These are both normative studies. They're both looking at development, but in this context, we did not explicitly examine development, different processing pipelines, and different acquisition strategies, as well as some generalization to non-human primates, which I will not talk about today, but an important replication in a single subject in using the myconnectome. And so to generate these MSNs, we take a multimodal data set like the NSPN project, or even just T1 features from from the NIH data set, which would only comprise the gray matter and curvature features here. And we look within a subject across parcels, and we normalize each of the features, and we look at the similarity profiles between the individual features within a subject. And it's a very simple method that we converge on for numerous reasons, and I can answer questions about the specifics of that and why we chose the different steps that we did later. And then basically what you get is an individual profile in terms of an adjacency matrix of interregional similarity across these different features within a single subject. And this is what a single aggregate feature average in this case across each edges in the adjacency matrix looks like for one subject in that data set. And so the top row, I'm also showing multiple subjects, 4 different subjects in this case from the NSPN sample, which basically shows that there's some consistency, but also inter-individual variation. The group average map is plotted here, and this is what we'll use to take forward into our gene expression analyses, and is generally kind of capturing the most variation in terms of the differentiability between lower order sensory regions and higher order association regions. And this is Russ Poldrack's average morphometric similarity network across his 14 different independent sessions. And so as an intro to the gene expression analysis, we aggregated the regional profiles of MSNs across different cytoarchitectonic domains, as noted by Solari and Stoner, which was a summary of von Economo and Koskinas' work. And it's important to show that in this case, the MSNs were capturing some aspects of cytoarchitecture insofar as the average similarity between regions within classes 1 through 3, which have a expanded supergranular layer structure relative to other classes, and is a source of cortical-cortical connectivity. And so this is also represented at the edge level, where we look at the highest value connections in the MSN networks relative to canonical structural covariance networks of cortical thickness, as well as diffusion tractography. And we showed that as you decrease the threshold and you allow weaker edges into the picture, that across all of those thresholds, the values that are captured by morphometric similarity occur between regions of the same cytoarchitectonic class relative to other regions, and also relative to the other group-wise networks. And so we wanted to further investigate this phenomenon a bit further with the Allen Institute data, and we looked at this in terms of a network-based statistic, a correlation across edges in this case, rather than across regions or samples. And to do this, we created a gene co-expression network where each node is a region, and each edge is the correlation value across about 20,000 genes. And we related that network to the group average MSN. And there was a strong relationship there. For edge level statistics, this R value is quite high. And to generate ranks for the genes to see how much they contributed to that relationship, we left a gene out, recomputed the gene co-expression matrix without that, and looked at the how the correlation shifted. So this first bit is to show that the human supergranular enriched genes, which was a set of 19 genes shown relative to mouse that the human V1 expressed, that basically the average expression of those 19 genes was higher in classes 1 through 3 of our cytoarchitectonic parcellation, which was a similar profile to what we saw with the MSNs. And these genes were defined by contributing specifically to the layer 2-3 expansion in humans relative to mouse cortex, although this was only in visual. The study was only done in visual cortex, but this pattern was specific to these supergranular enriched genes relative to similar sized sets of 19 genes randomly sampled in the Allen data. And we did some similar gene enrichment analyses, which showed some interesting enrichments, although a bit difficult to interpret because they were not very specific. As you can see, they're actually quite overlapping to what I showed you before with the scaling map, and that's also because the MSN map is correlated to the scaling map as well. It's independent insofar as we reran these analyses with brain-expressed genes only, and we found the same results. And we also ran this analysis where we controlled for total brain size in the creation of the MSNs, and we also see the same results. And so the final kind of validation that I wanted to talk about today was a recent study that just got accepted, and it has to do with validating the theoretical framework for how to investigate, or for why we would want to investigate normative gene expression profiles and integrate that with our brain imaging data. And so all of this, all of what I'm about to tell you comes from a small literature and one seminal paper that described this transcriptional vulnerability model, which states that the pattern of brain disruption occurs along the axes of normative gene expression. And so this was demonstrated in the context of Alzheimer's disease in 2009, showing that the gene expression changes across multiple regions followed a similar pattern as the number of plaques in the Brock stage of the disorder. And so in practice, what that means is that based on the normative patterns of expression for these genes, you could predict the progression stage of Alzheimer's. And so this was somewhat validated in recent studies by Jorge Sepulcher in Alzheimer's, relating amyloid propagation and tau propagation scores in patients to relevant gene expression profiles in the Allen Institute data, which is a normative gene expression data set. And so what we wanted to do was we wanted to test this in a more quasi-experimental way based on some findings that from Sarah Morgan and Ed Bullmore's group and other studies across different psychiatric disorders that also show that this transcriptional vulnerability model might hold insofar as brain-related changes indexed by neuroimaging follow along gene expression gradients in the normative Allen sample of, in this case, schizophrenia-specific genes. And so to do this, we used neurodevelopmental disorders cohorts where we knew the genetic architecture in a causal way. And that's another way of saying that is we looked at across multiple CNVs where independent sections of the genome were either deleted or duplicated, and these patients develop large-scale brain abnormalities in terms of brain size. They have very specific phenotypes, and they also yet share consistent phenotypes in terms of reduced cognitive capacity and developmental delay. And so basically, what I'm showing here is the different samples. They are quite small in some cases because these are rare conditions, but just reiterating the point that we have both duplications and deletions where we can look at gene dosage effects specifically for the X chromosome, where we'd hypothesize that the pattern for adding an X chromosome would be the opposite as the pattern for deleting an X chromosome in terms of brain structure, which has been shown previously. And so in this framework, we created MSNs with T1 weighted data across all the different patient groups, and each patient group had its own control sample. And when we generated these MSNs, we took that average statistic that I plotted earlier in the NSPN cohort, and we looked at the differences across patients versus controls. And based on these statistical contrast maps, we ranked genes based on their spatial correspondence to these t-statistic maps. And our hypothesis was that the skewness of the distribution of the relevant chromosomal gene sets that were duplicated or deleted would be the most enriched relative to the other chromosomes or the other locations in the genome. And so that's exactly what we found. In this case, I'm plotting the effect map of supernumerary X chromosome dosage versus Turner syndrome or X monosomy. And you see that the enrichments for the X chromosome relative to all chromosomes are the most extreme, and they're on the opposite sides of the distribution for the two maps. These effects that the brain effects that I'm showing you are not independent of each other based on our partial least squares model, but the gene loadings are. So effectively what that means is that the maps themselves don't show as statistically significant anti-correlation as the gene enrichment does. And that's further shown here in terms of the brain maps, where you can see that there's some overlapping structure here, but we're still sensitive to the known CNV in terms of the gene enrichment. Statistically, these p-values are highly conflated based on the size of the CNV, but the trends are highly consistent across the different disorders. It's much clearer to see this when you have a full chromosomal duplication or deletion, where you have a kind of a given set of nulls in the rest of the genome. With the subchromosomal deletions, we took two permutation strategies where we took the same size CNV and we looked at the median rank of the median position of those gene sets in just the chromosome of interest and also across the whole genome here. And that's why you see these two different plots. But these two subchromosomal deletions so-called fell on the same side of the distribution as the X chromosome, X monosomy. And so we wanted to look at kind of further annotation strategies of these gene sets because there's so many CNV related genes, and we wanted to be able to narrow down our search space to a few candidate genes that might be relevant to the morphometric similarity architecture. And so we used some available data across multiple different single cell RNA sequencing studies where you can actually capture the transcriptomic profiles of single cells, and you can do this in multiple brain regions. And basically what numerous studies have shown is that different cell types have different levels of gene expression. And you can sort these cell types by their transcriptomic profiles. And kind of alongside this transcriptomic revolution in the gene expression world, we also saw this really nice study by John Murray's group where they looked at the first principal component of the Allen Institute data, and they showed this interesting pattern relative to the T1-T2 ratio, in that there's an intrinsic architecture of the gene expression that somehow relates to hierarchy, as they call it. And so what this was telling us was that there was some axis on which the spatial variability was occurring. And so what we did was we integrated the cell class specific transcriptomic profiles and looked at the average gene expression in the Allen Institute across those cell-specific genes. And we did this using, at the time, the 5 available studies. There have been more since then, which we should update. But basically what you see in this heat map is the aggregation or the co-localization of cell type specific marker sets in terms of their average expression profiles in the Allen Institute, regardless of what study you're looking at. And so we operationalized that by looking at the average expression across these different specific cell types using weights derived from PCA in terms of how close to the centroid did these specific cell types fall. And using that weighted averaging procedure, we generated cell type specific maps that you see here. This decomposition is was just for visualization. And so what this said was that PC1, this was also using brain expressed genes only, as was done in the Murray paper. And the PC1 of this space is highly similar to the PC1 of all of the brain expressed genes. And so what that tells us is that the underlying architecture of the strong first principle component is most likely driven by cell types. And we did some validation of this, of these cell types maps relative to maps of evolutionary, developmental, and allometric scaling, as I showed you earlier in the first study, as well as the Hill and Van Essen study. And we used myelination proxies with magnetization transfer from the NSPN study. And we also looked at big brain derived supragranular cortical thickness, infragranular cortical thickness, and respective densities. And you can see hotspots where predicted independent models of the respective underlying cell type was correlated spatially to those cell type derived Allen Institute brain maps. And so we took this a bit further because we had, we wanted to look at the specificity of the spatial patterns between different cell types. And we used the Allen Institute in situ hybridization data to look at canonical marker genes between astrocytes, in this case, and oligodendrocytes, in two regions that we found a dissociation in terms of our deconvolution strategy of the Allen data based on cell type. And so what you see here is a double dissociation between GFAP and MVP, which are two canonical markers of astrocytes and oligodendrocytes respectively. This is qualitative, but at least it was some initial evidence that at least part of the spatial patterning for these cell types was justified. And so we had picked out earlier CNV genes or we had determined CNV genes that overlapped with the cell types and were also significantly enriched in the respective disorders. And we wanted to pick out a couple of these examples to further validate. And in this case, we looked at NCAM2, which is a gene on the 21st chromosome and is differentially expressed in Down syndrome patients, as represented by two independent studies of Down syndrome patients versus controls, looking at postmortem brain tissue, as well as differentially expressed modules derived from patient tissue in Down syndrome by Ninad Sesten's group, which he did in this paper, also validate against cell type specificity. And so what we had was a single gene that we had found using our deconvolution strategy that was differentially expressed in independent patient tissue, and that was also cell type specific. And this is just a proof of concept in a single gene in a single disorder, and mostly a post hoc analysis, but we wanted to test whether our methods were sensitive to something that was predicted by actual gene expression in patients, in patient brain tissue. And so in summary, I basically showed you three different types of cortical neuroimaging maps. And in the middle case, we actually represented it as a network in relation to the Allen Institute data. And we took three different strategies in terms of the gene expression mapping from the Allen Institute data to integrate with our imaging. And then in the last study, we tried to extend some of these methods into the single cell domain so that we could look at gene sets that we knew were specific to actual cell types and determine whether those spatial patterns were related to relevant causal mutations as indexed by morphometric similarity in neurogenetic syndromes. And so with that, I'd like to thank my previous supervisors and current funding situation. And I'd like to also highlight that in, I guess, a couple months from now, Jake Vogel and Ross Markello from the MNI organized this educational course where we'll be talking about these methods in a lot more depth, along with myself, Kevin Anderson from Yale, and Kirsty Whitaker and Petra Vertes from Cambridge. And that's pretty much it.
Fantastic stuff, Jacob. Thanks so much. We have a fair bit of time for questions, so if anyone wants to get started, or you can raise your hand or chat me a question if you like. Maybe I'll start. One of the things that's always, I've always wondered about the MSN work is how do you, if you see differences in network architecture between groups or as a function of age or sex or something like that, how do you attribute those to a specific component of the network rather than, because you have those 10 measures. Can you dial it back and say, like, actually what's driving this network change is the two things at the extreme ends of this correlation, for example?
Yeah. So, oh, sorry, go ahead if there's a,
no.
Basically in the, I wish I had supplemental slides here, but in the most recent paper, we looked at two things. One was the relationship between the T statistics between patients and controls when you left out a feature and how they changed. And the second was whether, actually three things. The second was whether the individual features are, for each cohort, and the T statistics derived for that, so thickness, surface area, et cetera, whether those were also related to the gene expression profiles in the same way. And the third way is we looked at the relationship between the edge level statistics between patients and controls in terms of what was the emergent phenotype that contributed most to the spatial pattern. And so without going into further detail, what we found, the general pattern, what we found is that different features contribute to the emergent alterations that we described using MSNs. And it's not the case that for all the different disorders in that context, that thickness or gray matter volume contributed most. However, gray matter volume was the feature on its own that best also predicted the gene expression, although it wasn't fully consistent across all of the different conditions. So there's still a lot of work to be done in terms of which features are contributing to a given phenotype that you're looking at with the networks. And I think that my hunch is that there is, like, we probably could find a predicted relationship between what feature is driving which phenotype, but it's definitely not in the context and the methods that we've employed yet.
Okay, I mean, that would be exciting, kind of but it's all very exciting. So, but that's really great. Thanks. So there's a question in the chat that everyone can see. So I guess I can say the name. It's Anastasia Brovkin from Germany. And she asked, one, since you mentioned the von Economo data set, would your analysis benefit from a more fine-grained atlas parcellation? And two, which parcellation did you use in the hierarchy in the AHBA atlas? Third part of the talk.
So yes. So the answer to the von Economo question is definitely it would benefit from a denser parcellation. I guess there's a few different strategies in that. I'm not sure what analysis kind of question that was focused on, but yes, there's only 7 regions that we classified. Von Economo determined over 40 different cytoarchitectonically differentiable regions. We could use that classification rather than the 7 classification. we wanted to kind of go from our, and this is the answer to the second question, our 308 region parcellation down to 7 instead of 44 or 45. And so that 308 region parcellation is a sub-parcellation of the Desiccant atlas. So all the parcels somewhat confine their boundaries to the Desiccant. And all of that's available in either the supplementary material of the papers or the reference is available for the GitHub page. I can also send that along too.
Anyone else have a question? Okay, so I'm gonna ask one more. For the last project, for the last, the third part of the talk, I mean, that stuff's really fascinating. But in a sense, you kind of have like, I mean, it's nice because you have a well-known deletion or duplication basically all the way through. So how well do you think that would generalize, or have you thought about how well this would generalize to disorders that have a genetic component to them, but that aren't defined by, a specific deletion or duplication that's so, that's so specific?
So I would say that the biggest caveat to that study is that nothing is independent. So the specificity that we're talking about is not kind of fully captured by the assumptions of that word. And so that's one big issue, is that the standard for specificity is basically full independence. But as you can see from the maps from that paper, that is not the case. We found trend level specificity of gene set enrichment, where we knew which gene set to look for. And I think that's a huge distinction. So I would expect that for generally duplications and deletions, it might be the case where you could potentially, at the very least, predict whether it's a duplication or a deletion. But I think, and we had a conversation earlier this week about this with another group that's looking at CNVs. I think the better way to look at the CNV landscape is to use a much larger sample of subchromosomal deletions and duplications, and query that space to be able to have a generative model for CNV, just CNV occurrence. And so that's to say that like the model that we found was also in part because of the CNV size that we were looking at. So half of our disorders or more than half of the disorders were a product of a full chromosome being deleted or duplicated. And so that's not, that is not created equal relative to the other CNVs.
Which makes sense.
Yeah.
Another question from Nadia Bolstein. So for leave one gene out method, to hierarchically classify the genes by contribution strength when examining the correlation between MSN edge weights and gene co-expression, how do you account for non-additive genetic effects like dominance effects or epistasis?
That's a good question. I guess in general, no one's really examined additive genetic effects in the Allen Institute transcriptome data. I think what this is getting at is the idea that you can't look at single genes. And I think that by and large, that's a true statement. We know that there's like an intrinsic spatial architecture to the Allen Institute data. And we know that that architecture can be explained by a few principal components. And so effectively, genes are not independent. And because of that collinearity, there might be a reason behind that might be additive genetic effects such as dominance or epistasis, but it's much more complex than that, because there's also methodological issues such as whether it's a brain expressed gene or not, and also whether you're normalizing the expression or looking at absolute expression. So it's a complex question, but I think generally to answer that, I would say, yes, it's probably very difficult to make conclusions based on one gene. Like if you were to take one gene and correlate that with your imaging map, because they don't operate alone.
Okay. Anyone else have a question to conclude? Going once, twice. Oh, Ray, go for it. You have to unmute yourself though.
Hey. Sorry, unmuting myself. Okay, I had a question about the MSN work. So there was a slide I'm remembering where you show a few of the MSNs one at the group level, one with like Russ Poldrax, and some of the individual ones as well. And like you can see from that slide, they are, as you said, similar, but some variability across individuals, which you expect. But I was wondering how, even though the patterns are consistent, you interpret the actual correlations being pretty low on both like either the positive or negative ends. And if that came up in any like questions or reviews or anything like that.
Yeah, so it's kind of like about the actual magnitude of effect that you're. Yeah, so one biggest, like the biggest conceptual issue is the idea that there are negative edge weights, which are induced because we normalize the features before looking at the similarity. I've tried non-negative approaches to combining across the different features, and I think my biggest worry is that when I look at other approaches, they're highly similar to the absolute value of the kind of MSNs as we look at it. So therefore, it's conflating actual edges that we define as having low similarity. And so that's kind of a roundabout way of saying, like, this is the magnitude is really difficult to interpret. And it's akin to looking at a functional connectivity network after doing global signal regression. And so it's a relative correlation value.
Okay, cool. That makes sense. Thanks.
All right, anybody else? All right, so I'm gonna unmute you all so you can properly thank Jakob for putting that in this talk for us. And thanks again, Jakob. That was wonderful work. I really appreciate it. So, thank you all for